I'm Dr. Steliyan Tinkov, a Senior Pilot Plant Manager from Novartis Technical Operations and BTDM on manufacturing of nanomedicines, a GMP perspective.
Ladies and gentlemen, good afternoon. Today I will talk about the manufacturing aspects of nanomedicines. It's going to be a basic theory. Only a second. Oh, okay. It's going to be a basic theory. I hope this is not going to be a low ball to you. But there is one very important message about it. I will tell you what. Then, in the second part of my presentation, I will give you a couple of practical aspects from my own experience with a couple of phase 2, phase 3, or commercial products. We will end up with some outlooks and readings. So now it's exactly 22 years ago after the FDA have introduced the first nanotherapeutic drug to the market. And now, from today's perspective, we have two journeys industry and regulatory agencies are taking. The first journey is about nanosimilars, about generic drugs, and how to characterize their therapeutic comparability with the originator products. This is a very challenging question, but our talk will be about the second journey, how to deal with modern therapeutic products which appear on the market, which are new on the horizon, and how to deal with their high complexity. Excuse me, how can I get back? Okay. High complexity. They have a very complex mechanism of in vivo action and safety. The second thing is the manufacturing process of these products is very sensitive. So their quality is very sensitive to small, even minor changes to process parameters. And now we have to differentiate how to deal with these aspects in order to be able to produce quality every time in full for the patients. Now what we see is a very, very simplified diagram, a process flow of an example liposome product. So we can see here we have a few process steps, and whenever we have a nanotechnological product, it will combine a different set of technologies. So in the most cases, there will be no two products which have alike technologies. So, for example, we can use high pressure homogenization to reduce the particle size, but some other product may require to have a membrane extrusion. So, whatever the differences may be between two products, there are a couple of things which are similar to the manufacturing process of all of them. In the most cases, the manufacturing process, the compounding part of it, will take several days. So, for example, we can have the phospholipid blending for these liposomes, the raw liposome formation and size reduction on day 1. We can have the remote drug loading and purification on day 2, and the stereofiltration and aseptic fill and finish on day 3. The second point is that we have a huge amount of process parameters. For such a process, we may expect to have over 1,000 parameters, which we have to get in control. And now the question is how to deal with this huge variety of different technical and process parameters. Ladies and gentlemen, these are maybe the two most important terms in the manufacturing of nanomedicines. The critical process parameter, there is a very vague and very subjective definition for this term. And almost every company has its own understanding, but this is the most crisp of them. And this is exactly what we need to understand. The number of critical process parameters must be very short. We must limit our 1,000 process parameters down to the narrow, meaningful amount of process parameters, and we need to control these parameters very strictly to put all our efforts to control them and to get them reproducibly, batch for batch, in order to work in our quality into the product. The term of critical quality attribute, well, this is practically every attribute of our product, particle size distribution or pH, inside, outside the liposomes, which is significant to our safety, identity, and therapeutic efficiency of our product. Now we have a couple of examples. And exactly this is what our 1,000 process parameters from our example process is boiled down to. This is only an example and maybe a not exhaustive list, but at the end we should end up with somewhere between 5 and 10 critical process parameters and link them to the related critical quality attributes of our process. So for example, by changing the homogenization pressure when processing the raw liposomes to sized liposomes, this will affect a lot the particle size. And now, if we don't have these two bubbles in control, our critical process parameters and our critical quality attributes, this will affect down the stream all our therapeutic profile of the product. And if we don't get these parameters in control on time during our process development, this will also affect our clinical trial results. And this is where we get to the last slide of the theoretical part. Now, we have here the streamline of research from the idea, early research, proof of concept 1, proof of concept 2, development, clinical phase until the market. We have to take a look at this arrow here. The regulatory agencies, they also want to know. They want to know what we are doing. They don't want to take part of it. They want to give advices. They want to be a part of the decisions we're taking. And now there are two very crucial points here. The nanomedicine candidate at this very early stage, this is where we need to know exactly what is the structure of our nanoparticle and what is the manufacturing structure, the manufacturing process. This is the process design. Is our manufacturing process scalable? And this is the second milestone here we have, first in man. This is here. This is where we need to know exactly what are our critical process parameters and our critical quality attributes. Now we can step into a couple of practical examples. Is a critical quality attribute easy to define? No, it's not. We will take a very simple example of a particle size distribution. You can see the definition here. So it's a very, excuse me, it's a very generic one, but it is a list of value or a mathematical function which defines the relative amount of particles present according to their size. Now, what is the size of these particles? For QA purposes, I actually need one number and two limits. This is what I need. And I need to be able to compare the numbers of one batch to another. But now, the particle size distribution is a very complex parameter. And we need to define early enough which of all these parameters are relevant for our product. I can take, for example, the mean value, the mean average of my particles, and to say, okay, D10 is important for the in vitro release, and D90 is important, for example, for the filterability of my particles. Because if D90 is too large, The particles will not get through the stereo filter. Now, everybody of us knows the process called membrane extrusion. It's a simple, common process, very well used in the laboratory, and now we have to take a closer look at that membrane here. It's a consumable, so nobody pays a lot of attention to the quality of these materials. It's a consumable, it's integrated in the process, and it's disposed afterwards. But now, usually you buy a package of these membranes and you obtain a certificate of quality or certificate of analysis. And you have 3 lines about the product: the average pore size, average pore density, and membrane thickness. But now, is this enough to characterize this material? And this is a crucial material to the process. In many cases, the pores of these membranes don't have the pore geometry you expect. And this may also affect your process. Now, a very short sentence about the microbiology of our process. As we said, the process may take a couple of days. The germs, the microbes, are going to grow during this time. And even in very simple process like tangential flow filtration, we don't have to forget that microorganisms are also particles, and we may end up with a out of specification result. Ladies and gentlemen, thank you for your attention.